video, we'll be looking at the concept of DNA replication and how the DNA molecule replicates. So a couple things just to review. I think we all understand this sort of stuff by now. Make sure that you do understand that when DNA replicates, it is making a complement to itself. So when T is being replicated, it makes an A. T will never match with a T. G will never match with a G. G always matches with C. T with A with T, C with G. You get the idea. DNA replication happens in the S phase of the cell cycle. Remember that, just to review. When DNA replication occurs in prokaryotes have a single circular chromosome. We've talked about this before, this idea of theta replication, because it forms this thing that looks kind of like a theta here, and a Greek letter theta. You know, if you turn this the other way anyway, it kind of has this theta look to it. And that's just because the single circular chromosome usually has a single point of origin and replicates. In eukaryotic cells, however, because the eukaryotic cells are linear and they're so big, or the eukaryotic chromosomes, there can be multiple points of origin. Now, the point of origin is just a place where replication begins. And when replication begins, each one of those points of origin begins replicating. Now, how does that occur? Well, if here's our strand of DNA, the goal of DNA replication, again, is to make an exact copy of this strand. And so what we will end up with is something that looks just like it. But how does that work? Well, in order for that to occur, that strand has to split right down the middle. Those hydrogen bonds that are used to connect the A's, T's, C's, and G's are going to split apart. And then each side, this side, the left side, and the right side, each side is going to produce a complement to itself. What do I mean by a complement? Well, every time there's an A, a T is going to come, a C, a G, and so forth. The other side is going to do the same thing, so that when you finish, this original strand is identical to the two that were made from it. Now, make sure you understand that the original strand is gone now because it is contained in the two new strands, right? And so really what you have in these two new strands is you have half that is old and half that is new. This is called the semi-conservative process of DNA replication. Semi because it's half, conservative because something is being saved. It's literally saving half from the original strand and making half of a new strand. And so those two new strands are identical then to the original strand. How does this take place? Well, again, you have here these points of origin, and then at each point of origin, replication is going to be occurring in two directions. As you can see, this is kind of happening this way, and this way, and this way, and this way, and eventually every bit of it's going to get replicated. It's just a quicker way to do that. Well, what we're going to do is we're going to focus in on just this, actually going this direction. That little section is called the replication fork because it's a fork. It's a fork. Don't try to read too much into that. All right, and so here's a picture of that close up. So we're going to be looking at this. I'm going to draw a picture of it because it'll make it a little bit easier for me to do that. And there's a few enzymes and proteins that are going to be essential for this process, and so we're going to detail those now. The first proteins that I want to talk about are these little red dots, and they are called single-stranded binding proteins. And the idea behind the single-stranded binding proteins is they keep the two pieces of DNA from not combining again with each other. They make the DNA stable so that it's separated. I mean, you think about saran wrap, you're trying to pull saran wrap out of the container, it wants to stick to itself. Well, this is like keeping those two pieces from sticking to themselves again because those hydrogen bonds are so strong. The next enzyme that's involved is this little enzyme, I'm drawing it like a loop here, and it's called helicase. And helicase is unzipping the two strands of the DNA molecule in that direction. Now, why is the direction important? If we go back here, actually here, you'll notice that the DNA molecule is parallel to each other. It is parallel, but it's actually going, one strand is going in one direction and the other strand is going in the other direction. And so what we call this, we call it anti-parallel. Each of those carbons are labeled 1, 2, 3, 4, 5 on that deoxyribose. And so DNA can only be read in one particular direction. It can only be read from 5, this is the 5 carbon, to 3, this is the 3 carbon. It can only be read in this direction. 5 prime is what we say because it's just the name of a carbon, 2, 3 prime, just like you read the English language from left to right. Well, the other side then has to be read in the opposite direction because 5 is down here, 3 is up here. So how does this translate to this molecule? Well, this is the 5 prime end of this one and the 3 prime end. Well, the other one's in the opposite, right? So the helicase is traveling from 5 prime to 3 prime. The next enzyme that's going to come down is called primase. And primase puts down a little section of RNA primer. What do you use a primer for? Well, a primer says, or a primer's how you start painting or whatever. So in this case, the primer is going to be used to start this replication process. The primer is a molecule that signals this other molecule that's about to come in. The next molecule that comes in is called DNA polymerase. And DNA polymerase is going to attach to this primer, and its job is to make nucleotides, or to make more of the DNA, and it does that by adding nucleotides. What do I mean by that? Well, every time it sees a T, it adds an A. When it sees an A, it adds a T. C, G, G, C. You kind of get this idea here. Here's DNA polymerase adding the T to the A as it's making this new strand of DNA. And it's going to go in this 5' to 3' direction. And as it goes along, it is creating this new strand of DNA. This strand that is going from 5' to 3' is called the leading strand. It's called the leading strand because it's going at the same direction at the same speed of helicase. I'll even draw an arrow there to help you remember that. However, this other strand cannot do that. It cannot go this direction because it can't go from 3 to 5. It has to go in this direction. So it can only replicate a small portion at a time. And so here's how that happens. It's the same concept. Primase is going to put down a little strip of RNA primer. Polymerase is going to come and add bases to make this new DNA strand. Primase is going to put down a little bit of primer. Polymerase is going to put down the strand. It's going to open up, helicase is going to open it up a little bit more. There's going to be a little bit more room, and the same kind of thing is going to happen. Now, instead of having this one continuous strand, you have these fragments. These fragments have a name. They're called Akasaki fragments, named after someone. All right, and so those fragments obviously cannot be left behind. They have to be dealt with. And so there's another enzyme that's going to deal with those fragments. It is called ligase. Ligase is a triangle. All right, and ligase literally removes these RNA primers and stitches together those Akasagi fragments to make the strand whole. And that is basically the process of DNA replication. Now, there are a couple of other pieces that are going on here. I didn't give this strand the name, by the way, because this strand is slower. It is lagging behind this DNA strand. This one that has to go in the opposite direction is often called the lagging strand. So you have the leading strand and the lagging strand. And again, don't lose sight of the fact that the idea behind DNA replication is to make two identical strands of DNA, half of it being the old DNA from template and the other half being the new. So a couple of other enzymes that are involved in this process. Not that one, this one. If there's a problem, if there's an error that is made, there's an enzyme that is called NUCLEASE. And NUCLEASE goes in and basically cuts out the bad portion or the error in the DNA.